a great pleasure in many ways as I looked for the little factoids of his life to realize that I think his life actually holds a lesson for the students in the audience, and I thought I would draw out those lessons for you. The first lesson is that when he was deciding on where to go to undergraduate school, apparently there was this discussion as to whether he should go to California or to Wisconsin. And apparently his mother suggested that it should be Wisconsin. And he listened to his mother. So that's the first thing I want you students to take away. Your parents have great wisdom. You should make use of it. The second bit of information, which I think is very worthwhile, and this is more for our UG students, but our PG students may understand, is that as an undergraduate, he had an opportunity not just to to meet the professors in classrooms, but to actually work with the professor as a UG researcher. And this actually made a profound difference in his career path and career choice. And I feel that this too has a great message for our students. He received his PhD from University of Alberta, and then actually has been in Canada ever since, and in Toronto. Currently, he's in the Department of Medical Biophysics, University of Toronto, and director of the Campbell Campbell Family Institute for Breast Cancer Research. I have his honors are too great to mention, but I've picked a few out that I think capture the essence of the extraordinary success he's had in science. He's a fellow of the Royal Society. He's been elected as a foreign associate to the National Academy of Sciences in immunology, been awarded the Order of Ontario, and recently inducted into the Canadian Medical Hall of Fame. So with that, please join me in welcoming Professor Mack to HKUST, where the title of his talk is going to be Animal Biology, Putting Anti-Cancer Targets, Put the Cart Before the Horses. Please help me welcome Professor Mack.
I'd like to thank Carl and Joseph and Nancy for this kind invitation to come back to Hong Kong. What Carl did not mention was that I actually grew up here, but somehow ended up on the other side of the ocean. So this is a talk. I first gave this talk about 5 years ago in a course in Bar Harbor, Maine, where I was then rated by students. You know, students nowadays have so much power. They put a lot of people in uncomfortable positions. And I was one of them because after I teach at that course at Bar Harbor. It's a very famous course. Actually, I have a picture in my second talk where there were 12 students there and 3 of them went on to win the Nobel Prize. That was 1954. And that was Howard Tedman, David Baltimore, and Mike Bishop. So that's a very famous class. And I gave this talk, this first title. I thought I'll try it out on really smart students. And I got my ratings back. You know, my ratings was somewhere around 5, you know, being 10 and 0, but mostly zeros, a couple of 10s, so that kind of dragged it there. And I remember one of the students' comments was a total waste of time. So if you want to do something else, fall asleep, go to the bar, that's fine. Now, this is a kind of a very unusual title, and I hope that in time you will learn what I'm trying to say. Of course, put the cart before the horses is, in English language, of course, it's the wrong thing to do because you're not supposed to do that. So I like to maybe begin by quoting this French playwright who said, "Doctors pour drug of which they know little to cure disease of which they know less, into patients of whom they know nothing. Now, it's 400 some odd years ago, and unfortunately, it hasn't changed too much. And so that is, I think, the biggest problem we have dealing today. Now, I'm technically a little bit challenged. Pointer. The green one. Okay, so green one. So I was actually lasering myself. But that didn't stop doctors from trying to save cancer patients' lives. You noticed that actually the first chemotherapeutic drug was nitrogen mustard. You know what that is? That's chemical warfare agents. But that came from some observation that soldiers that had been exposed to nitrogen mustard had smaller lymph nodes. So you see how thinking actually did help. And of course, the reason why it's listed up here is because it worked, and for lymphomas. And that's the reason why it's up there. But you can see that by the end of the last century, it was pretty clear that there was not going to be a lot more chemotherapeutic agents because it's just toxic chemicals, but it worked and it had saved many, many lives. So here's my grandson, and I took a picture as he was hitting the wall, and I said, by the turn of the century, chemotherapeutic agents have hit the wall. So it is just pretending it didn't really hurt. Okay. Now, so the idea is if we know how cancer is caused, then obviously we can do better than chemotherapeutic agents. And for the younger people, in 1966, I was just leaving Hong Kong to go into America to go to school. And it wasn't so far long ago as far as I was concerned. But yet at that time, 1966, scientists did not know the cause of cancer. So there are these 8 Nobel laureates who said, we are so smart, forget it, we will figure it out. So they all gathered together in this place called Lake Constance in Germany, which they still do every year. You know, this last year was physics. And the idea is that they will solve problems that the rest of us couldn't figure out. But at the end of the 3 days dissertations and discussions and arguments, the conclusion was cancer was not caused by virus. Okay? Now, you can't be any more wrong than that. And even, yes, you can be even more wrong than that, is that cancer is not caused by genes, right? And so they come up with some cockamamie biochemistry statement, which, of course, I went to University of Wisconsin biochemistry. For me, it was really very enlightening to read, and I was just starting. And little do I know that these guys already declared that the cancer is the replacement of the respiration of oxygen in normal bodies by a fermentation of sugar. What the heck does that mean, right? So, but just be careful. Now, he also said that science progressed not because scientists changed their minds, but because scientists attached to erroneous idea die and are replaced. Okay. So Otto Warburg died in 1970. Okay. And 6 years later, oncogenes was discovered by Stahlin, Varmus, Bishop, and Vogt. And of course, that was Salk. And then subsequently, Bob Weinberg and a lot of people discovered other oncogenes. And these usually were just genes that are carried out in viruses by chicken and mouse viruses that when put into a cell or an animal cause cancer. So the idea is that these are the genes that cause cancer and they are the horses. So if you want to stop your neighbor who lives a few miles away to come to visit you because you really can't stand him, what you do is you shoot the horses while the cart's trying to get to your house. And so the concept that you shoot the horses to stop the cart. So what are the horses? The horses are the oncogenes and the tumor suppressor genes. Now, that's the first problem already, right? Because the tumor suppressor genes are lost in function. The cart is moving because the horse is gone. Now, how do you shoot a horse that's gone? No, you can't. But there are still a few of these, and you know, if you look at the picture as half full, the glass is half full, we've done a lot. Okay. Spectacularly, 1998, HER2 was actually, Herceptin against HER2 positive breast cancer was actually approved. Dennis Slayman and Genentech. And it remains the first one. You know, how in Chinese family, they always think the firstborn is the greatest, the rest just forget, right? And it really almost is that. Because after that, there was BCR-ABL, a Gleevec, and that was euphoria. I still remember about 15 years ago, everybody thought cancer would be cured. And of course, the number of targets that are in green, that are oncogenes, continue. But you can see that with the hundreds of billions of dollars that have gone in, it really isn't that much, is it? So if you look at the glass as half empty, you say, whoa, I think we're going to hit the wall again. Yeah. Well, I just wanted in this audience be able to say that sometimes you don't have to be spending $2 billion. You can be smart, and Professor Wang and Chen Zhu and others have figured that acute promyelocytic leukemia is a translocation between PML and all translinic acid receptor, and if you make them develop, differentiate, and then kill them with arsenic trioxide, you can have great results. So what's the problem? The problem is what Bob Weinberg said in a meeting in Brazil, which I was in, and he said there are now more paths to developing cancers than there are stars in the skies. So why are we making some progress? Well, we're making progress because there are some one-star, one-horse cancer, okay? CML, BCR-ABL, APL, I just mentioned, HER2 positive breast and gastric cancer. They are spectacular. But if your cancer is driven by a star that has disappeared, and it's one star, ovarian cancer, neuroblastoma, you are in deep trouble. Because you cannot shoot p53. Because p53, the loss is driving the cancer. And if you look at another devastating disease, pancreatic cancer, you have 4 major horses. 3 of them are lost of function. P15, SMAD4, P53. And the only oncogene is RAS, and after 30 years, we have not figured out how to target it. And then you come into this big slew of breast, lung, colon that have so many oncogenes and all intertwined. We are in trouble. So you go to major cancer meetings, ASCO, AACR, the keynote speaker, always end at the end, it is an era of personalized medicine. It sounds really good. Hundreds of millions of dollars now poured into. In Toronto, we're building a $100 million personalized medicine center. What do they do? We'll take the patient, sequence the whole bloody genome, not bloody because it's really a bad thing, or amplifications, epigenetic changes, blah, 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 And then you use drug combinations. You use the pathway knowledge, and you use intuitive deduction. Well, I would say good luck, because there are dozens of pathways all intertwined. You don't know whether to hit upstream or downstream because of positive and negative feedbacks. And every time you combine two drugs, you cannot ignore the toxicity. It's not only going to double, it's going to amplify. So comes my kind of feeble suggestion that maybe moving forward, in addition to doing personalized medicine, I always say this because I don't want to offend the big guys, okay? That we have to start thinking about shooting the cart. So what is the cart? The cart is the transformed state of the cancer cell as a consequence of the actions of oncogenes and tumor suppressor genes. I'm not saying oncogenes and tumor suppressor genes don't play a role. They brought us there. But we are there. And there are so many ways that can bring us here or there that maybe we should start thinking about shooting the state. Because the state are independent on the oncogenes and tumor suppressor genes. Whether you bring the cart here by helicopter or by car, or you have students drag it cart up here, it doesn't matter. It's the cart outside. We have to deal with it, and we deal with it today. So what are the categories? These are the three things: immune, metabolism, and aneuploidy. Okay? The immune system. They don't care. T cells would recognize any peptide together with the MHC that has the avidity and the co-stimulation, and as long as it doesn't kill the patient, it can work. Well, I didn't think that it could work until the last 3 years. Metabolism. Because at the end, the oncogenes and the tumor suppressor genes change the metabolism of the cancer cell so they can adapt to the new microenvironment or the environment of the signal transduction pathway. It's independent of the oncogenes, tumor suppressor genes. You can be RAS, you can be PI3 kinase. You got there, you there. And I believe the last frontier would be aneuploidy. Now, we're all sitting in this room with 46 chromosomes in our cells. Unless you have Down syndrome, you have 47, trisomy 21. Or you can have XXY, you can YYX. But children born with 3 chromosomes, 13 and 18, are born and die within 2 months. So in other words, we cannot deal with a cell that is more than 46 chromosomes during mitosis. And yet cancer cells routinely, 70% of have 70, 80, 90, 100, 120 chromosomes. How do they deal with mitosis? If we know how to target it, we may have an interesting chance. So the immune system, the immunologists know and have been swearing and have been screaming, yelling that these are the tumors, and within the tumor are that CD8 T cells. And more CD8 T cells, the better the patients will be doing. Okay. But immunologists have cried so many wolves about cancer that basically the cancer field ignored it. So completely by serendipity, our laboratory knocked out a gene with an unknown function called CTLA4 in 1995. And what happened is, you can see, this is the mouse, poor mouse, 1 week after birth. I didn't take a picture of 2 weeks because you could see these are CD8, CD4 T cells. They are certainly not the kind of thing you can accommodate any longer. And so that became the first immune checkpoint. So immune checkpoints means you drive a car. If this car goes down, you go put in your key, you turn on the ignition, because that key belongs to that car. That's the T cell receptor, because it identifies the T cell with the car. And then you push on the brake. No, you push on the accelerator. Accelerator CD28. Then you go. Your car goes a block. A block later, stop sign. You put on your brake. That's CTLA4. So if you remove the brake, you turn on the car, you push the accelerator, and doesn't know how to stop. So that ends up being this mouse. Okay. So now, we were not smart enough to think of this. Okay. I went to my boss and I said, if we do something about CTLA4, the T cells will go crazy, and maybe it can kill chronic viruses. I never even thought of cancer. Okay? And of course, full credit goes to this guy, Jim Allison in Berkeley, who published a paper a year after us in the same journal. And it says that in vivo administration of antibody CTLA4 resulted in the rejection of tumors, including pre-established tumors. And his last sentence was, blockade of inhibitory effects of CTLA4 can allow for and potentiate effective immune response against cancer cells. Okay. The immunologist was screaming and yelling. The rest of the world said, these guys are crazy. Okay. I can say that because I'm an immunologist. Okay. So this landmark trial ended. And indeed, 10 patients died very, very immediately upon giving CTLA4 until the doctors can figure out how to deal with the toxicity. So when you let the immune cells rage, this is stage 4 melanoma, the green line. And if you give it anti-CTLA4, not only is there an extension of a few months of life, but what is significant here, 20% of them have a long-term survival, which today those patients are out 12 years without any relapse. So everybody is now figuring out how to lift this up and how to lift that. And what do? You're going to start doing all the combinations, boosting the immune system without killing the patient. And then there is a second break, PD-1, antibodies against PD-1 or PD-L1. You can see here in these lung cancer patients, the tumor go away. And these are end-stage patients. So I think we now start thinking about this is a cart, right? The immune system never really cared. But ironically, it is personalized medicine because your CD8 T cells look at every single one of your T cells and say, are we personalized for me to kill you? And if we are, boom, you're dead. Right? So there is no such thing more amazing than personalized medicine, the CD8 T cells. So the T cells actually, first of all, immune system starts with inflammation, then you've got recognition, you've got activation, proliferation, homeostasis, and inflammation again. So this process actually goes. Now this is advertisement. This book just won the UK Association Best Biology Book for the year. So if you want to buy this for Christmas to one of your friends, I would actually autograph it. Okay, so I'm not going to give you an immunology lesson, but you can see that there are many ways you can still figure out how to give the immune system even more burst. In most of the situations, 80% of the patients do not respond to these checkpoint blockade. It's because the immune cells actually cannot get into the tumor, right? And so I think this whole process is very important. I will come back to it. I'm not going to waste time telling you the different 5 steps that you can actually boost the immune system to sustain it. Now, for autoimmune diseases, what immunology has been trying to do very successfully is to dampen the immune system, like rheumatoid arthritis, colitis, etc., so that your immune system don't destroy your organs. But here we're turning back the table around. For cancer and for chronic viruses, we want them to go to the limit. Okay, so let's now switch to metabolism. So as I said, the oncogenes and the tumor suppressor genes are the drivers, right? Because they, p53, make PI3 kinase, CYP1 alpha, RAS, etc. They drive a normal metabolic phenotype because the cancer cell has to change. In fact, it was first observed, of course, by Warburg, 1928. That cancer cells use more glycolysis than oxidative phosphorylation. I'm putting some of you to sleep because I'm studying biochemistry now for the next 10 or 15 minutes. This change is necessary for 3 reasons. Number one, to get more energy. Now, actually, for most tumors, it's not that much. Because for acute myeloblastic leukemia, where a patient can come in on a Friday afternoon into our hospital, 5 o'clock, patient walk in, acute myeloblastic leukemia, 15, 20 minutes later, it's diagnosed. An hour later, the patient's treated because the patient may not last the weekend. Now, that's kind of tumor that you really need a lot of energy. But you have stage 1 breast cancer, which divides every 100 days. ATP is not that important. Biosynthesis, they need a different set of metabolites. So I went to biochemistry. I was told there are 20 amino acids. Of course, there are now 21. Right? So you guys can figure out the number 21. And we were told that there are 10 essential amino acids. At University of Wisconsin, we were told, how do you remember? Private Tim Hall, phenylalanine, valine, threonine, TIMM, HAL. But for cancer cells, there are 13. You can see that. Cancer cells need glutamine, glycine, and serine. Those are not essential amino acids for your normal cells, but they are for cancer cells. Okay. And the last, perhaps least understood, is redox. Okay. When you're renovating your house, when your house is readapting to a new storm that has just did a lot of damage, you're going to have chainsaws, you're going to have people soldering, there's a lot of heat. That heat can burn down the house, and that would be the end of it. So what do? You actually now give it redox. Like sprinklers, fire hydrants, fire deterrents, etc. Okay. So, and one of the molecules is NADPH, right, which takes glutathione disulfide into glutathione. Glutathione takes your reactive oxygen. When I teach students, I'll say, what's the most abundant molecule in your cells? A few of them say actin. I say, hold a minute. Okay. And some say ATP. Okay. The smart aleck will say water. Right? But the real answer is glutathione. There is 10 millimolar of glutathione in your cell. What's that mean? It means this place is spinning with sprinklers non-stop every day at 10% of the volume of this room, of the number of items. So this is because the reactive oxygen generated by hypoxia, metabolic stress, ER stress, oncogenes, RAS activate, RAC1 activates NADPH oxidase. What does NADPH oxidase do? It only does one thing. One thing. It makes reactive oxygen. Okay. So this is going to burn it down now. So what do? Here's your glutathione, NADPH, NRF2. NRF2 is the master transcription factor for antioxidant. Couple dozens of enzymes require NRF2 to be activated as a transcription factor to take care of all the reactive oxygen, and then tumor suppressor genes and dietary oxygen. So if this doesn't come in, these guys are going to burn the cell to death. Just remember that. So it's actually quite contrary to what you're told. They say, take vitamin E to stop cancer. So we have a paper in press in Cancer Cell. I'm not going to talk about it, just coming out. It actually says that antioxidant gives you more cancer. And it's very simple thinking, because if these guys are burning cell down, you do not want to rescue a damaged cell, because the damaged cell is actually going to kill you later. Okay? So let's come back to NADPH. So it turns out whether you are pro-democracy or anti-democracy or pro-Beijing or a crocodile or a yeast, there's only 3 ways you can make NADPH. Pentose phosphate shunt, glutaminolysis, isocitrate dehydrogenase. The pentose phosphate shunt. Cancer cell turn on glucose channels. It goes from 100 to 20,000 as soon as you activate PI3 kinase. Because AKT, mTOR, HIF1 alpha, boom, all the GLUT channels. And glucose come in, pouring in. That's why you get a PET scan, right? But it turned out cancer cells then turn on pyruvic kinase M2 and put a stop, and this is discovered by Lou Cantley's lab. Well, it's actually discovered by a German lab who published it all in German, and so, that's too bad. And pyruvic kinase M2 shuts off the passage from fructose 1,6-bisphosphate and back it up to glucose 6-phosphate, go down to pentose phosphate shunt, where you get ribose 5-phosphate, other things, but you also get NADPH. Right? And Lou Cantley's lab brilliantly figured that out and published a paper in Science that the pentose phosphate shunt was an NADPH. Glutaminolysis. Children with acute lymphoblastic leukemia get treated with L-asparaginase. And you can see the percent of blast counts come down. Single agent, L-asparaginase. Now, do you not believe in cancer metabolism? It's L-asparaginase. We've known this for 40 years. But doctors always say, well, here it works. Don't ask me why. You figured it out, smart aleck. So, okay. So it turned out it was glutamine, the glutaminase in the L-asparaginase, as well as the L-asparaginase. If you take L-asparaginase without the glutaminase, it won't work. And the glutaminase kills off the glutamine, so the glutamine cannot go in and form a redox. Okay. So I'm wasting my time trying to convince you with what seems to be so trivial and so unbelievable, but I'm going to convince you of this isocitrate dehydrogenase. There are 3 isocitrate dehydrogenases, 1, 2, and 3. Don't talk about 3 right now. Just 1 is in the cytosol. It takes isocitrate to alpha-ketoglutarate. It takes NADP to NADPH. You get your redox. The second one, IDH2, it's in the mitochondria. You take NADP to NADPH. You get your NADPH. So, isocitrate converted to alpha-ketoglutarate by the cytosolic wild-type IDH1 And then alpha-ketoglutarate is involved in transamination, where you make amino acids, and from amino acids you make lipids, you make glucose, you make carbohydrates. Okay. So that's already about 50 reactions happening right here. Okay? And alpha-ketoglutarate is the key. Now let's add another 86. Another 86 reactions that are completely dependent on alpha-ketoglutarate. And these are the dioxygenases, alpha-ketoglutarate dependent dioxygenases. They are the histomethylation genes, TET1, TET2, which converts 5-methylcytosine to 5-hydroxymethylcytosine for epigenetic changes, porohydroxylation of HIF1 alpha, without which cannot be degraded by VHL, E3 ligase, collagen type 4, without which it cannot fold properly, so it cannot mature. So this little alpha-ketoglutarate sits in the middle of your whole metabolism. It's that level that changes everything in your cell. So cancer cell figured out too. So they mutated IDH1 and IDH2. Chondrosarcoma, 30%, cholangiocarcinoma, 30%, AML, 15%, gliomas, 90%. A little bit of everything. Myelodysplastic AML, IDH2, angioimmunoblastic T-cell lymphoma, which our laboratory, together with the French group, discovered. They went out, mutated this gene. And when these genes are mutated, the usual suspects of oncogenes and tumor suppressor genes go out the window. Here's primary glioblastoma. You have 23% PTEN mutation, 38% EGF mutations, PDGFR mutations. Secondary glioblastoma, 0, 0, 85. And yet, if you take two samples of primary and secondary glioblastoma to the world's best neuropathologists and say, tell me which one is which, the honest one would say, no, I can't tell. The dishonest one would say, yes, I can tell. And then you say, okay, I've got 50 more here, and blind test. And then he's dead or she's dead. Because you cannot tell the difference. So what does this do? What does the mutation really do to this? So isocitrate dehydrogenase 1 and 2 takes isocitrate to alpha-ketoglutarate. It turned out the mutation is a neomorphic enzyme. In other words, it has not seen the germline of animals in the history of this planet. It takes alpha-ketoglutarate, which is the product of the wild type, and take it down a different path. That path lands up in 2-hydroxyglutarate, which you do not have to be a biochemist to see the structures are almost identical. So when this is present at a high level, up to millimolars in the brain of the tumors, and up to millimolars even in the blood of patients with acute myeloblastic leukemia, they block the alpha-ketoglutarate functions. So all these, in addition to the metabolism, all these 86 alpha-ketoglutarate dependent dioxygenases for cytosine demethylation, chromatin structure, collagen maturation, and HIF1 alpha degradation, etc., are altered by this 2-hydroxyglutarate as a negative allosteric or enzyme, probably more at the enzyme site blocking the reaction. So our laboratory, Sasaki Masato and Christiane Knopp, who, by the way, is a German brain neuropathologist, and she told me that anyone who thinks that they can tell the difference, make them do the test and bet them a really expensive bottle of wine. You always win. Okay. So we knock in these mutations in the brain and the hematopoietic tissues because those are the most significant tumors that we're dealing with today. And just one slide for the brain. And this shows you how effective it is. So if you knock it into the brain, what you do, you see that there is collagen type 4 maturation defect. So when your collagen type 4 cannot fold properly, you can imagine something can go wrong. Okay? I can't imagine what it is, but it is. And because the porohydroxylation HIF1 alpha is not happening, the E3 ligase VHL cannot degrade it, and you accumulate HIF1 alpha. And HIF1 alpha is considered an oncogene. Okay. So that's a little bit more difficult to figure out, but this is an easy one, right? If you knock it into the hematopoietic tissue, what you see is splenomegaly, extramedullary hematopoiesis, increase in progenitor cells, decrease in bone marrow cellularity, anemia, and if you tell that to a hematopathologist, he said, that sounds like myelodysplastic syndrome. That means it's pre-leukemia. Right? So I just want to show you a couple of slides that hypermethylation changes because of the mutation and for the same reason that I already described, because it is involved in activation of TAT, which then leads to cytosine demethylation. And the changes that you can see in these hematopoietic tissues in this mouse is like at the p-value of 10 to the minus 36, similar to acute myeloblastic leukemia patients' changes. So that says its relevance is very high. So the idea is that because of all these metabolic changes revolving around a simple molecule called alpha-ketoglutarate, that leads to the hematopoietic stem cells increase in symmetric cell division. In other words, it's more likely to make one of itself than to make two of more mature progenitor. And then as a long time passes, you accumulate a lot of these progenitors because of these metabolic changes, and then you come secondary tumorigenic hits. You get FLT3-ITD, you get RAS mutation, you get WT1 mutations, and then you get acute myeloblastic leukemia. So what's the proof? Well, the proof is to take the IDH1/2 knock-in and breed it into another knock-in like FLT3-ITD or NPM1 or RAS or whatever. So Evan Lind, who's now a new staff position in Oregon Cancer Center, he did this experiment. He took an IDH1 knock-in at 3 months, FLT3-ITD knock-in at 3 months, but if it has both, it has frank leukemia, acute myeloblastic leukemia in 3 months. Okay. So, and just to be more dramatic, the mouse dies. Okay. So, it's done this, and it's done this, you're now here. So the question is, has the boat leave the harbor? In other words, are the boats gone? There's nothing you can do anymore. So They have IDH mutation, they have FLT3-ITD mutation, they have RAS mutation, they have this mutation, that mutation. They're gone. Inhibiting the IDH no longer bring the boats back. So a company in Boston, Agios, made an inhibitor to isocitrate dehydrogenase 2 called AG221. By inhibiting this, you drop the alpha 2-hydroxyglutarate, and this blockage is then released. Can you save the patient? So 30 patients were treated up to about 2 months ago. This just to show you how dramatic this is. The first 10 died before they got the dose. That's how fast these refractory acute myeloblastic leukemia. 9, upon given the inhibitor, went into complete remission. In other words, the doctors say less than 5% blast. 5 went to partial remission. In other words, it's somewhere between 5 to 10% blast. And these patients started with 80, 90% blast. Interestingly, 6 couldn't care less. They just smiled, continued to go on and kill the patient. Okay? Now, it's very interesting to look at the data. It is not the usual let's kill the cancer cell. In fact, it did not kill the cancer cell. All it did was to drive the leukemia blast from the bone marrow into the periphery. You can't stay here. It's like in Canada, in the middle of winter, in January, in the middle of the night. You don't like your friends in your house? Kick them out. Tomorrow morning, you call the ambulance. They're dead. These are mature granulocytes that coincidentally peak as the blasts come out. In other words, you change the bad guy into good guy. You did not shoot the bad guy. He became a good guy and he died a good guy. How much more can you ask? So it's really very interesting because if you now start thinking what another drug called BTK inhibitor is doing to chronic lymphocytic leukemia, you see that these patients have lymph nodes swollen to the point they can't even talk. You give them the BTK inhibitor, Bruton tyrosine kinase inhibitor. Here, did you see? Same thing. All the blast came from the neck and the lymph node into the blood. But this is a different situation. It's in Siberia. It's a Russian guy who got kicked out for being drunk, and they go and die out there. So we're talking about a new way of treating cancer without killing them. Because we just drove from the chronic lymphocytic leukemia with BTK inhibitor, what we know is the calcium signal. You kill the calcium signal, they can't stay, they gotta go. And AML, now we gotta figure out how this works now. The IDH inhibitor did something and they went from the bone marrow into the blood and they died there. A natural death. And the BTK inhibitor is, of course, a very important calcium signal. And so it's a very complicated story. I think it's different time. You invite me, we can come back and talk about it. So let me now move to the last 10 minutes and to ask if reactive oxygen, which is, nobody wants to hear about it because it's too complicated, can it be used to answer some other questions? For example, patients with BRCA1 mutation have 70% chance of breast cancer and 40% chance of ovarian cancer, and nothing else above background. Why? BRCA1, same function in every cell. Why do these poor women only get breast and ovarian cancer? And the guys carry the mutation, get away with murder. Because nothing does anything to them. And for 20 years, nobody figured out why. Okay? So came Italian postdoc called Gorini. She deleted BRCA1 and showed reactive oxygen shoots up. Okay. The reactive oxygen shoots up because the master transcription factor of NRF2 is down. So if it's down, that means the fire hydrants and the sprinklers are down. The same fire would have burned the cell down, and the cancer cell dies. If it happens in lung, liver, kidney, colon, that's the end of it. You remember, these individuals carry a bad copy of BRCA1. When you lose your second in your lung cells, they die. Okay? So she did really a spectacular experiment. So she actually took a p53 cell and deleted BRCA1, and instead of the mouse tumor getting more tumor, the tumor went down because BRCA1 protects NRF2. So when you delete the second allele, you cannot protect, and the cell died. Now this is really the kind of story you tell your grandchildren. She discovered when the mouse was pregnant, the tumor came back up. So she did the ultimate experiment, took these cells, implanted into male mice. Of course, they won't grow. And then she gave the male mice estrogen. So in other words, when you delete the second allele of BRCA1, ROS goes up because NRF2 goes down, the cell dies in any tissue except breast and ovary. But in breast and ovary, you have estrogen. It comes in through the PI3 kinase pathway to rescue with NRF2, and then you rescue a damaged cell. The damaged cell without DNA repair, it's a pretty bad situation. And that leads to p53 loss, p10 loss, and mutations, you get cancer. So what's the proof? Well, actually, these patients, 95% of them get a very special kind of very, very bad breast cancer called basal subtype or triple negative. If that's the case, the basal subtype of triple negative should have a decrease in redox, because you can only partially rescue it, but an increase in reactive oxygen. Okay? So you go to the database and look at every single database and ask if the triple negative basal breast cancer have lower level of redox because of this model. And the answer is yes, yes, yes. All three databases show a decrease in NQO1, which is an NRF2 target. So what we're saying is that there is these patients who lose the second allele of BRCA1, they ordinarily would have died because they have NRF2 this low. But if it's breast and ovary, estrogen comes in and rescue NRF2, you get to live. But you get to live in a damaged situation. And not only is it damaging your DNA, it's damaging your DNA repair genes. 42% as opposed to these, 24% as opposed to these. And together with ovarian cancer, very similar picture. That's why they also get ovarian cancer. P10 in this context is involved in DNA repair. So that leads to the oxidative stress going up, DNA repair going up, and you get aneuploidy because your chromosomes are just breaking over and they're translocating, and it leads to chromosome instability, genetic aberrations, and malignancies. That is why the basal subtype of breast cancer has this profile. Very, very damaged. It's like it's been through hell. But the nastiest criminals have been to hell. They can do the worst things to you. And that ends up being highly aneuploid, triple negative. And so the question is, what are we going to do about it? So it turned out that aneuploidy is known at low level to drive tumorigenesis because you keep breaking all the different chromosomes and translocating. But can we actually push it to high aneuploidy and kill them? In other words, how do you fight a burning oil well that is burning out of control? You bomb it. So what can we do to change aneuploidy from low level to high aneuploidy and get a bad cancer to die? So we don't know. So why don't you take 10 breast cancer cell lines here, 10 breast cancer cell lines here, and you screen 9,000 genes and ask which gene when knocked down kill these and not these. Don't be greedy. Don't take those that will kill both, because those that would kill both will kill you. Two genes came out, polokinase 4 and TTK. What are they? Polokinase 4 is in the centriole, counting the number of chromosomes per centriole. It makes sense, because you're an aneuploid cell. You need more of this enzyme to count. It's like when there are 23 pairs of twins going to school, at the end of the day, there are 2 cars coming back in the school bus, and in the middle time, the 180 kids ended up in the school. How do you repackage everybody back into the bus? You need special teachers, and you need special ropes to bring them back into the car. And the TTK is a spindle assembly checkpoint. So if you knock down PLK4, the cell dies. So we raised some money together with Dennis Slayman, who discovered Herceptin, and then we did some chemistry in China and in Toronto. We ended up with a molecule which is pretty potent. It's about 0.6 nanomolar. 0.6 nanomolar. Okay. And it completely does not touch PLK1, 2, and 3. Okay. So when you kill PLK4 tumor, guess what's happening? These are the centrioles. So in other words, when you have the inhibitor, they cannot count. And you kill it with TTK, they cannot proceed with their chromosome segregation. So luckily, we filed an IND, and it was approved in the US and Canada, and we treated our first patient in April. And this is completely different from any of the other mitotic kinases. But what is the amazing thing is, at any concentration of this drug, no cells have 2 centrioles or centrosomes. If you have no 2 centrosomes, you cannot divide properly. If you 1, you're dead. If you 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, you're dead. So, and the mechanism which it works is actually amazing. The enzyme degrades itself upon autophosphorylation. When you block that, the enzyme keeps going up. The enzyme drives aneuploidy, as was told to us by David Pellman at Harvard in a 2009 paper in Nature. That PLK4 at high concentration drives aneuploidy. So the inhibitor, in essence, is creating an in situ increase in PLK4, which will then drive itself from low level of tumorigenesis to a high level of aneuploidy. That results in the drug being highly effective in a lot of tumors. And this is an ovarian patient-derived xenograft, never seen plastic. And the mechanism by which it works is my second last slide, in case you're falling asleep. It's time to get up. That the carboplatin resistant ones, you can see about 10-fold in each pair. This doesn't care. And certainly, if you take a triple negative breast cancer, you get in the beginning, they never recovered. Here, the blue is 75 mg per kg of carboplatin, which is the mainstay of treatment of triple negative and high-grade serous ovarian. Completely oblivious to carboplatin until you hit it and it comes down. So that's the end of my story. I need to thank these people who made the isocitrate dehydrogenase work, Masato Sasaki, Christiana Knopp, Rob Karen, Stefan Gruth from Agios, Maria and Ari from New York. Maria is now in Michigan. Evan Lind is now in Oregon. Pam Ohashi, Francois Lemonet and Philippe Goulard in France. Kiara and Mona for the BRCA1 work. And these are the doctors and the chemists who are leading the chemistry work and running the clinical trials. And thank you very much. Any questions?
I have a couple of questions. The one is on a second strategy on inhibitor of IDH2. Is it targeted? Does it target to only the mutant form?
Yes, it does only target the mutant.
Mutant form is enough difference.
Yeah, it's actually the drug has so far not reached maximum tolerated dose because it's a compound that hits an enzyme that doesn't exist in your body,
Okay.
except in the tumor.
Mutant form. Okay, the next one is the last part. When you did this RNA screen, you knocked down the gene
Right.
for the effect, and but the drug you said increases stability.
Yeah, when I knock down the gene,
Increase Aneuploidy.
either way is killing the cell. So it's a very interesting, I'm glad you pointed out. You're the only one that's not sleeping. Okay. So blue here is 1 centriole. Okay? Green is 3. Okay? And then purple is 4, and then 5. So you see it goes both ways. It goes from, now this is a normal, it's mostly 2 centrioles, the cell goes divide. At 10 nanomolar, you're seeing that most of them now, or at least half of them, have got 3 centrioles. These cells are not going to survive. Okay? And then at 50 nanomolar, they get 3 and 4. These cells are not going to survive. At 100 nanomolar, which would be the optimum, basically there are no 2 centrioles. Okay? Now all of a sudden you move to 200, you go to 1. So the knockdown and the inhibitor are flipping this one way or the other. So it's actually a very, very unfortunate thing for us because less enzyme is bad, more enzymes is bad. Less enzyme, it cannot split. More enzymes, it cannot recluster the centrioles.
So then follow-up is what happened to the other cells with the same treatment and in this range of dosage?
Normal cells?
Normal cells or what the other type of?
So it turns out that it kills 50% of the patient-derived xenografts, and the other 50, they don't care. So I think the idea is if it has a little bit, and you inhibit it, and you chase it to kill itself through aneuploidy. And if there isn't PLK4 is a very interesting enzyme. You can knock out the gene completely, and the mouse lives day E7.5. That means it blasts through gastrulation without the function of this gene. So we don't really know what this gene is doing, except it sits in the centriole.
So you examine the effect of these drugs on a normal kind of breast cells?
Yeah, we've done through mouse, rats, dogs, everything the FDA wanted. Now we're into patients, and I don't know the results.
So, based on the IDH results, then, you would almost predict that nutritional therapy, altering either, does it work to use altered nutritional levels of alpha-ketoglutarate, or my favorite amino acid, glutamine, to either enhance these studies or, get after the cart in a different way?
That's absolutely excellent question. I wish I can make a prediction for leukemia, because we have no idea what's going on. But in the brain, your favorite amino acid is number one, right? Because your glial cells, which by the way is 90% of your brain, almost 90% of people think 90% of your brain are neurons. No, you're not that smart. You're mostly glial cells. They rewire the whole Krebs cycle to make glutamate becomes glutamine, then pump it out to the neurons to suck it up and change it into glutamate and change it into GABA, and you got your cognitive functions. So I think your favorite amino acid is dead on. We just begin to try to look into that. In hemopoietic system, I don't know how that is going to function. Maybe, do you think glutamate is involved in hemopoietic cell differentiation?
No, I think glutamate might be.
Yeah.
Driving the same, through the same metabolic pathways that you were, because it was one of the three entrees into the Krebs cycle you were talking about. Yeah.
Well, that's why L-asparaginase work for the kids.
Right.
In fact, it also, I asked doctors why it doesn't work in adults. Occasionally, L-asparaginase, I've seen patients, not my patients, where acute myeloblastic leukemia patients with 50% blast taking L-asparaginase, the blast went away, single agent. Okay? And they all come back. So the explanation is that we are exposed to the L-asparaginase comes from a bacteria throughout our lives. So we have antibodies against it. So that's now, I don't know, but this is very intriguing.